No, we share this vision that through a smarter organization, we can really speed up drug development. But something's holding it back. If we consolidated knowledge and data, we would be able to make better decisions. And if we designed our workflow around what we already know, we would be able to get more things done. So why aren't we?
Casper, why don't you introduce yourself?
I certainly can. I'm Casper. I am a entrepreneurial spirit who has worked ever since my youth on a sequence of startup companies, the latest one being Abzu, but also by far the one dearest to me being Abzu. I have worked mainly with high-performance computing and data analysis for the last 25 years in different contexts. That kind of thing has now acquired the name artificial intelligence. It's still data analysis. And here at Abzu, we are taking a leap into a completely new approach to data analysis that I've been thinking about for more than 25 years.
And you found yourself in the life sciences sector, maybe for the first time, is that?
That's the interesting thing about life, and I guess startup life, is that you never know where it takes you. I had no idea I'd end up in the life sciences or in pharma. Actually, I'm on the physics and mathematics side of things typically. But I've worked in many different industries, from shipping to marketing to all sorts of industries in my career. But actually never life science. It's a new one for me. But it is a place where analyzing data makes a lot of sense.
Yeah, for sure. I've been in life sciences for all my career, in fact, and so I'm having the slight switch in terms of working with such interdisciplinary people. I've dabbled in that, but we're part of a really diverse group of people and great diversity in skill sets. I was kind of pondering on how I arrived here a little bit when we were planning this discussion, and I was thinking, when I was an undergraduate, there was no such thing as the human genome. It hadn't been sequenced, and that happened about midway through my PhD, which was in life sciences, and I felt something seismic was about to change in the way that we approached drug discovery, new medicines, understanding diseases, and that was 20 years ago. And now I think we'll probably touch on the phrase omics, polyomics, multi-omics. So that was the genomic era, and now we're in the multi-omic era, which, as you've alluded to, involves lots of data. And I think pharma, for one, biotechs, the whole life sciences sector are realizing how important it is to embrace that, to embrace the wealth and diversity of data. But there are problems there, and maybe we'll talk about some of the problems around data in pharma and in life sciences. Do you have any initial thoughts or comments on what the big issues are around the...
So let me first say that I'm with you. I was also apprehensively waiting for the human genome to be sequenced back in the zeros. Even though I wasn't in life science, I'm a curious person, so I was following it carefully, just like I'm following everything that the James Webb Telescope is doing these days. And I had the same feeling, not as informed, I'm sure, as where you came from, but back then I had the feeling that life is written in a programming language called DNA. So now that we have reverse engineered that programming language and laid it bare in front of our eyes, we would understand. I wasn't naive enough to think that we'd understand all about life and how it evolves, but I had thought that we would understand more than what has come out of sequencing the entire genome. And I think from a information theoretical point of view, I was surprised, but I shouldn't have been, thinking about information theory as it really is, and how complex systems interact. Of course you couldn't explain biology from the DNA. But that just means that we didn't get the benefits that we immediately thought we'd get, but we got a lot of very interesting things to study and try to understand that it's leading its way into new science. And for me, being part of that new science, it is a scientific frontier in a certain sense to understand what kind of data we should collect, and what data means what, and what data in a certain sense even is when you're talking about biology. It's extremely fascinating, and it's right where we are.
Yeah, it was 20 odd years ago that the human genome was published, and we were just having a conversation earlier today about the fact that 80% of that was kind of thought of as junk, just carrier DNA, not doing anything. And it's 20 years on, we're really starting to get a better understanding about why there's 20% that's really important, but 80% is highly involved in intricate regulation of that 20%, and it opens up the whole drug discovery sphere, really, to think beyond the 20% that makes a protein that you can target as a drug, which I think is the sort of traditional drug discovery route that people have taken, making small molecules or antibodies to these proteins, and 80% is still unexplored or just starting to be explored.
Yeah.
And I think that's kind of mind-blowing.
There's a lot we could say about that, right? So first of all, yes, even the term junk DNA, from a certain perspective, I can see why you would come up with the theory that the stuff that gets converted into protein is the stuff that matters, and the rest is just there for evolutionary leftovers. But on the other hand, in any system that you study, this is maybe the physicist speaking, in any system that you're studying, there are no inconsequential components, right? It doesn't exist. It can be that the consequences are hard to detect, or maybe they're not particularly big in a given context or in a given time frame, but there are no components to a physical system that have no consequences. And therefore, of course, the junk DNA has consequences, although not as linear and directly as perhaps the coding part of the DNA. But I think that's also what you're pointing to here. That most of drug development today, as I see it at least, and of course you're the bigger expert, but as I see it, most of drug development, because we're using antibodies and small molecules, really are limited or constrained to targeting the stuff that gets turned into proteins, and that cuts off a lot of causal threats or avenues of action in the organism that we just cannot attack with those kind of drugs, because it's after it gets transcribed to proteins. Yeah. So what treasure trove of potential targets, really druggable targets, lies in the 80%, and that of course brings us to RNA therapeutics that we both care about deeply. I know. Yeah.
Maybe it's a good time to go into the RNA therapeutics world. I did research before moving into business development and before abzu, and I heard from pharma companies time and time again, we need better disease understanding. We need academia to find us new targets, because there's lots of money in drug development, but we're all chasing the same targets, and that's a phenomenal waste of money. It's not serving vast numbers of patients for which there are diseases with absolutely no medicines on the horizon. and things just need to be changed. The whole way about tackling drug discovery is a bit outdated. And I think the RNA therapeutics revolution that is starting is one way of opening up those opportunities. And I would say one of the beautiful things about RNA therapeutics is that it marries itself so well with omics data, sequences. These drugs are precision medicines. They're designed down to single nucleotide precision, and combining understanding genetic evidence, proteomics, all these different omics, being able to put them together and start understanding what's going on will lead to thousands of new medicines for thousands of diseases that as yet have got no treatments, and I think that's really exciting.
There is still the topic about. So one thing that when I first learned about RNA therapeutics, specifically antisense oligos, I was, well, here's a silver bullet. You just design the reverse transcript of the whatever mRNA or pre-mRNA you want to knock down, and voila. And then I realized, right, but that requires it to be stable, to be deliverable into the nucleus in this case, to have no adverse side effects of any kind, to be perfectly safe, and of course those are the other details that hides the devil. And in that sense, I think RNA therapeutics is in its nascency. There's a lot of things we don't really understand about the way it also impacts the body, other than its direct impact for which it's designed to. So that can mean that you get this feeling of there's a long way to go, but for me it's more like it's a treasure trove of undiscovered discoveries, and that fascinates me in the field. There's also another thing about why I'm really fascinated with RNA therapeutics as a modality, which is that it opens the opportunity for the scientific method being applied for target discovery, right? Because what's so hard about target discovery is that it really is just a hypothesis, right? You dig and you study and you read and you measure and you analyze and you compute statistical probabilities, and then you end up with a belief that this target plays a certain role in the development of a disease. But in honesty, it really is a fairly unsubstantiated hypothesis at this stage in almost all situations. So for a scientist, I think the next natural question would be, what is the easiest possible way I can falsify this hypothesis? The answer is right there, right? RNA therapeutics can readily test your hypothesis, at least if you're able to express it in a way that can be tested in a cell line. But no matter what, it is the easiest way you can possibly think about to validate or falsify a hypothesis about a target playing a certain role in a biological mechanism. And that makes it a wonderful tool and a very underutilized tool, I think, in the industry.
So we have been talking about the traditional way that drugs have been developed in pharma and biotech, and that being a very linear process. So you have an unsubstantiated target, you try your best to validate it, and you'll do that in vitro, in cells in a dish, and the ones that still look good then go into small animals and then to bigger animals before they ever get to clinic to test the first in man studies, as they might be called, phase 1 studies for toxicity. That is a very long and a very expensive journey. I think the attrition rate is something like 80-90%. It's and the cost to go from idea to testing in clinic to drug on the market can vary traditionally between $1 and $10 billion. It's insane. And so if 90% of your drugs are failing, something needs to be done. And we have the idea, I think, collectively, that with the information that we can gather from these RNA therapeutics, and with the data that comes out, we can start to integrate all the information that we're getting and feed it back continuously, which we are terming a drug discovery loop, an omics drug discovery loop, odd loop.
I can jump in and say, again, as an outsider coming to the field less than 2 years ago, I knew next to nothing about disease mechanisms and how the drug development process actually worked. So there was two things that surprised me, and therefore also two things that I think it's important to tackle, and I also think it's possible to tackle them. So, and I think the best way to phrase it is, or at least for me, a good way to phrase it is in the context of another field that I worked a lot in software development, where when you develop software, you want to make better decisions about what to try, and you want to fail faster with the things that are wrong. That's really the two pieces. You want to get more software out the door, more workable, usable, nice software out the door. You try to become better at making good decisions about what to build, but acknowledging that it is just speculative, you also try to find out how can I fail fast if I'm not building the right thing. Yeah. Now, translate to drugs. It's a scientific, it's decisions about what to try, what to do, driven by actual scientific knowledge, is the make better decisions part. Yeah. And finding a way to test the things that you believe but which you're uncertain about at as early as possible a state where failing is cheapest is the other part. And I think maybe when you say the odd loop, that's what you mean. Yeah. You mean being able to make informed decisions, also make informed decisions about how to test what you still don't know, test what you still don't know, and then bring that back particularly when it fails. Then you don't progress in the loop, right? You go back, but now you go back with knowledge that makes you take more informed decisions in the next try. So ultimately the loop just spins faster and faster because the knowledge builds up. Yeah. But in the knowledge build up is the reason for there's something about how you organize yourself to achieve this faster loop, which is cross-functional teams and empowered teams and those kind of things. But that's one part. The other part, the thing that spins the wheel faster and faster, is actually the accumulation of knowledge.
Well, that's an interesting point. So individual entities, organizations, pharma, biotech, could be doing this themselves and keeping what knowledge they have to themselves. But there is a wealth of information globally, and if there was confederated data of some description, would that make everything work? Most likely.
It's one piece, right? But ultimately, it's not about data. It's about understanding. Returning to the spin faster and faster metaphor, it's the understanding that makes the wheel spin faster and faster. Yeah. It's not the data, but the data can facilitate the understanding. It doesn't come naturally. Actually, that's where data analysis comes in, right? Data analysis and scientific analysis of data is about converting data into knowledge. Mm-hmm. So if you take only the rapid accumulation of knowledge part of the challenge, yes, data is part of it, but analyzing that data in appropriate ways and interpreting what comes out of that analysis and committing it to your understanding, to your common domain of understanding, is equally important. Yeah. I'm a huge fan of the idea of consolidation of data in various ways. Mm-hmm. in a federated setting where data stays where it is, but you can learn from it in a distributed fashion, or in a centralized setting where there's some kind of shared access. Either way, what you get is the ability to extract knowledge out of the data. But even if we fail at that, all hope is not out, because if you can extract knowledge from the data, then that knowledge can be committed to the shared pool without the data ever being shared in any meaningful sense. I think in a way that's how the history of science has actually progressed. Sometimes we repeat each other's experiments, but most of the time we build on top of each other's conclusions and understanding. Yeah. So there's a differential learning in that sense happening in science as such, which I think is also the hope here.
Casper, we've come together today to talk a little bit about what's exciting us in drug discovery today, and the roles that you and I may play in it, but we want to dig a little bit deeper with domain experts. And so what would be top of your mind in these conversations?
I don't know. We share this vision that that through a smarter organization and through a better utilization of data, more scientific, more knowledge-driven decisions, we can really speed up drug development. But something's holding it back. Right? That's why it's interesting to talk to people out there in the real world being held back, because it's not what we're looking at here. It's not rocket science. It's pretty clear that if we consolidated knowledge and data, we would be able to make better decisions. And if we designed our workflow around what we already know, we would be able to get more things done. So why aren't we? Is it? And I have a couple of thoughts that could be interesting to hear if people agree that this is the problem. So one problem certainly seems to be that much of the drug development process today is structured around the reality as it once was, where it was incredibly hard to validate your hypotheses along the way. So how do you actually invalidate whether a target that you're hitting with a small molecule is a good target, right? It's a long way before you know. You can't really test it in a single cell. It's very difficult to actually do that. So you probably structured the workflow around the reality of, say, small molecule drug development, which doesn't really translate that well to particularly not to RNA therapeutics, but most likely not to peptides either. So that's an assumption, and I would actually really like to have some people reflect on that, people who know this industry from the inside. Yeah. Another thing is perhaps about cross-functional barriers, like a medical doctor and a chemist and a statistician perhaps not speaking that well together, and often being siloed off in different departments. So this cross-functional superflow that we're looking for is very hard to make happen. Maybe this is a bigger thing in big pharma companies and much less so perhaps in smaller biotechs. But certainly that's something it would be interesting to hear from people in the industry. And finally, and this is a technological piece, that's why I came to this field. I think that most what people call machine learning and artificial intelligence systems today are simply not up to the challenge. They are designed to predict stuff, not to explain stuff in any meaningful way. And what we're talking about here is accumulation of knowledge. And for predictions is not knowledge, explanations is knowledge. So maybe there's a technological barrier that has prevented the industry to take this direction. And it would be very interesting to me to hear from the actual practitioners in the field to what degree they feel this, to what degree have what I perceive as shortcomings in neural networks, for instance, in that they are essentially black box predictors or behave like the Oracle of Delphi. Once they've trained, they just throw out wisdom with no explanations. To what degree does this actually contribute to this less than optimal organization of drug development? So those are some of the questions I think it would be interesting to dive deeper into with real practitioners in the field and not just an outside observer like myself.
Yeah. Great, we'll endeavour to do that.
Sounds good.
Give each other a high five.
Oh dear, sorry.
Whatever is it.
Yeah.